So yes, the presentation today, based upon our long work in the area of adaptive optics microscopy. And what we're actually trying to do here is come up with, for what's happened over the last 10, 20 years, is that many people have been working in the area of trying to implement adaptive optics in microscopes with many areas of success. But until now, we don't have a universal solution to this. There are many, many different ways of doing adaptive optics in microscopes, and no single one of them has solved all of the problems. And part of this is to do with the complexity of this problem. There are many different types of optical microscope out there. There isn't just one optical microscope architecture. And because of that, there are many different types of adaptive optics methods out there. And what we've been trying to do as part of the work over the past few years is come up with something which will become much closer to a unified solution. And this is really what this talk is about. The first half of the talk will be giving a lot of background about this problem, and the second half of the talk will be talking about a particular framework we've now put in place based around machine learning in a way which will be slightly unusual, which we'll explain to you. And most of this machine learning work has been sort of implemented by the first two people you see on this list here, Chi Hu and Martin Hailstone's very good ideas about how to actually implement this. And so I acknowledge them at the beginning and the other authors about how we've actually put this together. So as a background to the research group here, we cover a wide range of areas of applications of imaging and adaptive optics. And you can see them summarized on this slide here, ranging all the way from things like neuroscience imaging through to laser material processing and even optics for space applications too. But a lot of this is centered around the idea that we can do adaptive optics and use adaptive optical elements to improve the properties of these optical systems. And indeed, a lot of it to do with improving them in microscopes. And that is the topic of what we're covering today in this talk. And so you can see that over the years, we've developed numerous different ways of implementing adaptive optics in different microscopes. Just 6 examples shown on this slide here, ranging from what you might call conventional resolution microscopes at the top through to super resolution microscopes at the bottom. And I could make this slide, put a lot more onto this slide, including our work and work from many other groups as well, which have been done in this area, to show you the large volume of interest in this area and the large number of applications. So let me first, for those of you who are not so familiar with these applications, explain to you the background to what we're doing here. And so what we're doing is we're using reconfigurable optical elements, dynamic optical elements, to introduce equal but opposite aberrations to those which are introduced by a specimen. So we could use either a deformable mirror or a spatial light modulator to do this. Now, a deformable mirror is a reflective membrane whose shape we can change by controlling voltages applied to electrodes. And the whole idea here is that the light, if you think of light, a collimated laser beam or collimated light beam reaching the deformable mirror, then if you can change the shape of the mirror, then you can change the shape of the wavefront which is reflected off the mirror, as illustrated in this diagram here. And this also works in a reciprocal direction. You could have a distorted wavefront approaching the deformable mirror, and the shape in the deformable mirror will remove that and turn it back into a collimated wavefront beam and a flat wavefront. So we either use a deformable mirror or we use a spatial light modulator. And the spatial light modulator is a liquid crystal device which operates on a different principle to the deformable mirror, but has a similar purpose. We can control the voltages across each of the pixels of the liquid crystal device and change the phase of the light and the phase of the wavefront which reflects off the spatial light modulator. And so again, very similar idea here, different mechanism within the device, but again can be used to impart a phase variation across a wavefront, or indeed in the other direction to remove a phase variation from a wavefront. So this is what we'll use for controlling aberrations in our microscopes. And we will build our microscopes in this way here, where we will place in an adaptive element at this stage of the microscope. So if we're thinking of a laser scanning microscope, we can think of the beam coming in, being pre-aberrated, where we pre-shape a wavefront so that when it passes through a specimen, now this specimen here is going to introduce some distortion, an aberration, which is going to cause problems in our imaging. But if we can pre-shape a wavefront so that it compensates whatever was introduced by the specimen, then we should end up with a diffraction limited focus. Now, also, this would work in the other direction. If we have light emitted from the specimen, it would pass through the upper layers of the specimen, become distorted, and we could use the adaptive element to remove it. And so we would use either the deformable mirror or the spatial light modulator in this position here in order to do this. And we, depending on the configuration, we might use a spatial light modulator. It'd be well suited, for example, to a laser illumination path, whereas a deformable mirror is much better suited to use in, say, fluorescence microscopes, where you're using broadband and unpolarized light. So that's why we might use one or the other of these. And some of the examples you would see is we've used either or both, depending on the configuration of the microscope. Now, the most likely source of aberration in a microscope comes from refractive index mismatch. That is when you have materials which have different refractive indices. So, for example, there would be the immersion medium of the objective lens. So typically we use immersion objectives, so using water immersion or oil immersion or something similar, in order to get higher numerical aperture and therefore higher resolution out of the microscopes. But there may be a mismatch between that medium and the specimen mounting medium, or of course the cover glass, which you'd often have on the microscope specimen. And these aberrations due to refractive index mismatch increase with depth, they increase with the numerical aperture, and also to do with the refractive index change. So normally you would think of a focused laser beam being focused down by a lens, and the rays would meet at a point, or if you did the ray optics modeling, you would see the kind of distribution we see on the left here, where the optic axis goes from top to bottom on this diagram. And you see that just by the nature of focusing, the point spread function of the microscope would be elongated along the optical axis. But you would think of this as a three-dimensional focal spot in a microscope, because most of these microscopes we use to get three-dimensional image out of. Now, the problem is that when you have a refractive index mismatch there, such as is shown in this diagram, you see that the rays will be refracted at the surface. Then they will no longer meet up at a tight focus. And if you do the modeling of this, you will see that you end up with a highly distorted focal spot. It's elongated by the spherical aberration and it's lower intensity. So we end up with a loss of resolution and a decrease of contrast in the specimen, in the image. But refractive index isn't the mismatch, this mismatch between two refractive indices. This is not the only source of aberrations in biological specimens. We also have the refractive index variations throughout the cells and throughout the tissues. And so many years ago, we did an experiment to measure this, where we took laser light, we passed it through the specimen, and then measured in an interferometer what the phase of the wavefront would be. And then we go and focus through at different points within the specimen, and then look at what happens in the pupil plane, and this where we measured the phase. And this is what we see as we focus to different points in the specimen. You can see that the phase of the wavefront changes. It changes from different parts of the specimen. It varies in shape and size. And so you see clearly here that if you want to go and image different parts of a large specimen, you will have to change the aberration correction. So you will need an adaptive form of aberration correction in order to make sure you get good quality imaging at all positions and at all depths. So conventionally, originally, adaptive optics was introduced for astronomical telescopes and is indeed widely used there. And so one might think, well, can we look at adaptive optics and astronomical telescopes and use those in microscopes? Now, typically in all the examples I show here, and almost all astronomical telescopes use adaptive optics, they have the configuration shown as here in the top left, where the light coming from a distant star, either a natural star or an artificial guide star, would have an aberrated wavefront. That would be coupled off into a wavefront sensor, which would detect those distortions, and that information would then be used to control the correction element, which would remove the distortion in the wavefront. And this light would then go on to form the image of the camera of the telescope. Now, this method of using wavefront sensor is sometimes applicable to microscopes, but not always. And it's to do with the difficulties of being able to measure wavefronts when you've got a three-dimensional object and you've got light coming from many points within that object. And so rather than, although this can be used in certain microscope configurations, we've developed other methods based around what we call image-based or wavefront sensorless adaptive optics in order to control most of the microscopes we've implemented. And the idea behind that is summarized in this slide here, where what you do is you would take a sequence of images where for each of those images you have applied a chosen aberration. So that image will be aberrated. They will have its original unknown specimen aberration and then whatever you've added to it. And that aberrated image will contain some amount of information which you can use. And you'd be able to calculate from that how good is the image. You calculate some form of quality metrics such as image intensity or image contrast, which tells you how good that image is. And then you would go and take another, choose a different aberration, apply it to acquire an image, and calculate this. And then you would cycle through here, taking a collection of images. And from that collection of images, you would then estimate the correction phase. Now, one way in which that works is you can have, we understand how the aberrations affect the imaging properties of the microscope. So by changing the amount of aberration mode we apply, we can actually come up with a metric function like image intensity, which is we know is maximized when that particular aberration mode is corrected. So we could take a sequence of images, from that sequence of images, project what the best correction is. And then we could cycle through the modes, and as illustrated in this diagram here, do it on a mode by mode basis, taking, for example, aberration modes like astigmatism or like coma, and going through them and correcting them in order to get the fully corrected image. And we put a lot of work into understanding the mathematics of these processes in order to optimize them and make them work. I won't go through all of the details of that in this talk, but I will just give you one example to show how this can work. In this case, what we've got is a microscope. It's actually a transmission microscope, but we've taken initial aberration here of this scattering plate. And you can see that it's, you can see some features in it, but it's also a little bit aberrated. We then apply a positive amount of the mode we wanted to measure and a negative amount. We take these three images. Now, the collection of these three images, you can see there is some information you can derive about the relative qualities of these images. And in fact, we know this mathematically, we know what the function is. And so we can use a quality metric to work out points on this curve. And then because we know the nature of this curve and the shape of this curve, we can then work out where the optimum point would be from those three measurements and correct it. And that's the principle of what we're doing here, but expanded up into many more aberration modes. And we've developed these methods for different types of microscopes. So just give you two examples on the next slide. The one on the left is where we've used 2-photon fluorescence laser scanning microscope in order to look at these are fluorescently labeled mouse embryos. So this is like a ball of cells, approximately 100 micrometers thick. And we're focusing through to the bottom of this. And you can see that in the top without aberration correction and the bottom with aberration correction, you can see that we've been able to create much sharper images with features, more visible features in there, where you can see, for example, cell boundaries, and you can see much more detail in these. These are actually the DNA of some dividing cells, which have been labeled with the fluorescent dye. And this has all been done by using this image-based optimization. The example on the right is a much more recent one where we've applied this to a wide field sectioning microscope, and the colors in here represent the three different fluorescence channels we've used. So, and you can see in this case that we've also been able to use image-based methods to correct and reveal much more details in these areas of these specimens here than was present before we corrected the specimen-induced aberrations. Now, these methods use, as I mentioned before, there are different types of microscopes. These two particular types of microscopes work on a very different basis. They also had different designs of the adaptive optics scheme, image-based adaptive optics scheme, in order to make this work. And this is something I'll be talking about later, is how can we use a system which doesn't require specific pre-design of this, but perhaps learns how to cope with the differences between microscopes. And that's what we'll be looking at later. Now, aberration correction is not the only thing you can do with adaptive optical microscopes. And so before I move on to the later parts of the talk, I'll just give you some examples of other things we've done by putting adaptive optics elements into microscopes. So one thing you can do is we can use a deformable mirror, not just to correct aberrations, but also to refocus the microscope. If you put focusing can be affected by putting on a quadratic or an approximately quadratic phase onto the pupil of the objective lens. And so you could use a deformable mirror very easily to implement this effect as well. And this is useful because in many applications, you want to rapidly focus through to different planes of a specimen and get a volumetric image. Now, how this is usually done is by moving the specimen relative to the objective lens. So either moving the specimen or moving the objective lens. And this is difficult to do at high speed without severe agitation of the specimen. And so doing this optically with a deformable mirror enables you to do this faster without disturbing a specimen. And so this has been implemented in this particular case to do volumetric imaging of a fly brain. So the video I'll show you here, where you can see at the top the green slice here, this shows 5 slices being quickly acquired from this specimen, and you'll see two things. One thing is you'll see this jittering around, and that's because this fly is alive and we're observing its brain while we're actually alive. The other thing you'll notice is that it's flashing. It's flashing because the fly is being exposed to different odors, and the neurons in the fly are labeled with a calcium sensitive fluorescent dye, and this dye lights up when the neurons are activated. So if you I restart those. If you then run this experiment, different neurons fire when the fly is exposed to different odors. And you can see from this map at the bottom, this color-coded map, we can extract from these videos how the different collections of neurons fire simultaneously depending on the different odors, which are encoded by the abbreviations IAA, MCH, and OCT, as shown in this graph here. So we can get not only the spatial distribution, this also the temporal distribution of these signals. And so this is only possible because we can very quickly refocus using the adaptive optics. Another area of a totally different way in which we can apply adaptive optics is for imaging inside mouse brains. In this particular case, we can think about using, normally you would think about using an objective lens, but you can see this is the size of a mouse brain here. And an objective lens is very big in comparison to this. So you think if you want to go inside this, then this is not very effective. Another way people have actually tried to get down to deeper levels inside the mouse brain is by using a graded index rod lens, which is shown here. And in order to do this, though, you have to remove a lot of brain tissue and disrupt the brain function in order to be able to do this type of imaging. So because these things are 1 or 2 millimeters across. However, if you were able to image through a multimode optical fiber, which is 1 to 200 micrometers across, then you have a very thin needle-like probe, which will create a lot less disruption to the actual tissue if you wanted to image deep inside this. And many groups have been looking at how to use adaptive optics to do this, and there's a real problem because when you fire laser light through a multimode optical fiber, you get something like shown on the right here, which is basically a speckle pattern. The light is scrambled through the multimode optical fiber, and you can't really create an image out of it. However, it is possible to use adaptive optics because if you use a spatial light modulator and you can measure the correct or obtain the correct phase pattern, you can apply a phase pattern on the spatial light modulator which is conjugate to the phase which will distort, which is distorting the cause the distortion through the fiber. And if you can determine and apply the correct phase pattern, you can actually turn this speckle pattern into a single output focus. And if you can then change an appropriately trained sequence of phase patterns, you can actually scan this spot around and then create a scanned microscope image. And many people have worked in this area, and certainly not the first people to do this, but what we were able to do was one of the first experiments using this to actually create images in a live mouse brain. And so with collaborators here in the Department of Pharmacology in Oxford, we were able to do this. And here you can see this is first on excised tissue, but you can see that we can get, using the multimode fiber adaptive optics method here, we can get similar quality to what you would get out of a conventional confocal microscope. Perhaps more importantly than this example in a fixed specimen was actually being able to do this in a live mouse brain, where the fiber was input through to the lower layers of the mouse brain here, and we were able to actually image fluorescently labeled neurons, and indeed see these spikes on the side are boutons. There are the processes coming off the side of the neurons, which we can resolve and observe at different depths over time in this mouse brain. So whilst there's still a lot of challenges to make this a universally applicable method, we can use adaptive optics as well to focus and do imaging through these multimode fibers. So those are just some examples of how we can use adaptive optics to improve microscopy methods. So I wanted to talk the rest of the talk about this universal framework we've been coming up with to try and enable us to have some method which can be translated across different microscopes and across different specimens. So just going back to this slide, which you've already seen, this is the basis of what we're doing. We're trying to take a sequence of images with different aberrations applied, and then from those images estimate what the correction phase should be. And fundamentally behind this, there are some choices we need to make, which are essentially where these highlighted green boxes are. And what we can say we're trying to do here is we're trying to, in our overall goal, get an accurate estimation of aberrations over the largest range of aberrations we can, that's both complexity and size, with the fewest image measurements. And the things we can control in order to do it, so the ways we can think about the aspects of this algorithms include the aberration representation. In other words, which phase pattern should be applied when we take these measurements? What is the optimization metric? How do we extract the information, useful information from the image in order to optimize this problem? And once we've done that, what algorithm do we use in order to do the estimation? And to take one example of the first example of this, the aberration representation, let's look about what that could be. Well, we've done investigations in this area and discussed, well, what are the optimum shapes? We typically think of these things in terms of Zernica polynomial modes. This is just Zernica polynomials are what most people use to model aberrations. So that's usually what we've used to do this, but it's not necessarily the best way to do it. And in fact, if you look in the literature, you will find that people have done image-based adaptive optics using other modes as well. So one thing we did a couple of years ago was try to do comparisons between different methods of image-based adaptive optics. And we took different methods where we either use Zernica polynomials, which are defined over the whole pupil as shown here, or methods where we can split up the pupil of the objective into different regions, either a large number of regions where each one of them is just moved by a constant phase, or a smaller number of regions where we have a small number of aberration modes such as piston, tip and tilt. So these are just different ways of dividing up the aberrations either as modes or as zones or as combinations of zones and modes. Because these, in the literature, you look around, you think, well, each one of these has been presented, each one of them seems to work. Every individual author seems to give the impression that their method is better than everyone else's. We all do this. This is normal in science. We like to promote our own methods. But how do you compare these? Is one actually better than the other? So in this particular paper, this reference at the bottom, we went through and we tried these out. We did side-by-side comparisons to see, was there a big benefit from using one method or another method? And we found that, well, in general, all of these methods work. In some situations, one method might work better than others. So, for example, in this one here, with relatively simple aberrations, we found that the modal Zernica methods work well, whereas the other methods are much slower to work, but you just need more iterations to get them to work. So there are various questions you can ask about what is the best aberration representation, and we started trying to answer that there. Another thing, the next point we had on our list was what's the optimization metric we should use? In these particular examples, we were using a 2-photon microscope. We just used an image intensity to do this. It's a very good metric as a proxy for quality of image or correction of aberrations. But in general, across microscopes, you need something a bit more sophisticated than that, something which maybe looks at image sharpness, looks at other image features. So we also looked at that, and one thing we came up with was using wavelet decompositions of the images. Now, if you're not familiar with wavelets, then what they are a little like Fourier decomposition, but also incorporate both spatial frequency and space information. So you get local features, but also you get to know something about their spatial frequency distribution, their sharpness, if you like. And so if you look at different bands within a wavelet decomposition, you see different features of the image. And one thing we realized was that when aberrations increase in a specimen, you get a transfer of information from the higher spatial frequencies to the lower ones. So from some of these wavelet, for example, these wavelet coefficients through to the next ones. And so the transfer of this energy between these bands of wavelets actually gives you an idea of how aberrations are being corrected. But importantly, it will give you information about different degrees of aberrations. So if you've got very small aberrations, a very small amount of small aberrations will start removing your higher spatial frequencies. Then you move up to larger levels of aberrations, they start removing the larger spatial frequencies and so on. So in the different in the different wavelet bands, you get different information about different degrees of aberrations. And we can use this information collectively, not just for a single optimization metric, but a form of joint optimization metric. And that's what we implemented in this particular reference here, where we were able to, the reason why there's several curves on here is because we were doing a joint optimization of all of these bands. And this turned out to be very useful in some forms of microscopes where, for example, an example of a super resolution STED microscope here, where depending on how much you can basically tune the resolution of the microscope, and therefore you would tune the effect different aberrations have on different effects. So what we learned through this process was that simply choosing an optimization metric like intensity is a bit limited. So we started thinking, well, how do we know which is the best way to design this? How do we take this knowledge from these particular steps and then come up with something which actually finds the optimum design for us? And this is why we moved on to thinking about using machine learning to do this. And so what I'll show up in the rest of the talk is an implementation where we've replaced these more conventional algorithms I'll be talking about here with a machine learning based algorithm, which is inspired by and takes into account the things we've learned through this process. And I think that's a very important thing to point out in this case, is that there is a lot of machine learning work going out there in microscopy. 99% of it is talking about post-processing of images using potted and rather complex neural networks. What we've done here is a little bit different, as I'll explain. So here's the overview of this. I mean, this is basically summarizing very similar to what we had before. We start off with an aberrated microscope. It's got an adaptive element in it. We then have the correction mechanism, which actually is very similar to the, in simple design, to the flowchart I showed you earlier. We apply aberrations using the deformable mirror. We take a sequence of images. Then we will calculate, using the algorithms we developed, the correction. So in many ways, all we've done here is changed slightly what we were doing before and swapped out the conventional optimization algorithm with a machine learned optimization algorithm. The rest of this process is the same to what I said before. But behind that, we want to try and achieve certain things. So the desired features of this method, we want to make more efficient estimation of aberrations from fewer images. We'd like to use better use of image information. So using something more like the Wavelet decomposition metrics, but perhaps even better than that. We'd like to, if we can, increase the estimation range, the size of aberrations we can estimate with these, because most of these methods were relatively limited in range. We want to have robustness to microscope variation. So if we switch between different microscopes, we can still make these methods work. Now, this is a very important one, trainable and synthetic data. Most machine learned methods based around imaging, are based around using massive data sets. We're talking about millions of images, typically. You can't get that. You cannot do that in a microscope because it would take weeks. It would burn all your specimens. It would be a disaster. You just cannot do this. You might get thousands of images. You're not going to get millions to do this. So really, you need something which is trainable on synthetic imaging models which model the imaging operation of the microscope. We need to be able to use that. And that's an issue because it's very possible in these things to overtrain things to a particular model, whereas actually you need to cope with the natural variability between microscopes. And that's related also to the translatability, the idea that if we invest effort into this, we want it also to be applicable to different types of microscopes. And I think to a large degree, we've already managed to achieve most of these points. So, as I mentioned before, you look in the literature about machine learning assisted aberration estimating, or indeed any machine learning assisted methods in microscopy, and most of it's based around deep convolutional neural networks. And I've just highlighted this particular paper here because I feel I can, because I was an author on this paper, and so I feel comfortable putting this one up and pointing out the deficiencies in it. But this applies to most of the other ones out there as well. You have a very complex neural network in here. It takes in images and tries to estimate what the wavefronts are. And what you find out is that, well, one good thing is it was trained on synthetic data. That was one of the points we wanted. But you find out this is a complex network, an incredibly complex network with many parameters, thousands, hundreds of thousands, if you like, of parameters in there, which is suspicious because when you actually think about the maths behind this problem, it's not that complicated. Secondly, it still had a highly constrained practical range. So it didn't, we could reconstruct wavefronts, but not over a very big range. It relied upon point-like objects. It relied on bead images to do this, which is not what you normally have in a microscope. You normally have extended objects and specimen structures. And it was highly specialized to one system. So this didn't meet most of the objectives we've stated on the previous slide. But whatever new method we come up with should meet those objectives. Trained on synthetic data, it should be simplified network. It doesn't need to be that complex. And we do this by making sure we use our physical knowledge before we design the neural network to do this. We want a larger operating range. We want it to work on arbitrary extended objects, and we want the concept to be translatable across microscopes. Now, the way in which we do this is this physical knowledge thing here. We should take a step back, look at what we know about how microscopes work, how the mathematics of image formation work, and then use that to turn this problem from a complex one into a much simpler one. And this is what we do here. We do this by data pre-processing. So let's take a really simple example here, where we just take two images out of our microscope, and in one image we had a positive amount of astigmatism, and in another image we had a negative amount of astigmatism. We end up with two images from our microscope, each of which is aberrated. This is what we were kind of doing before in our sensorless adaptive optics anyway. But what we now do is we remember that these images are a convolution of the sample with a point spread function in both cases. And the point spread functions in these cases will be different because we've applied different aberrations. So what can we do with these two images? Well, if you take the Fourier transforms of these images, these convolutions, as you all, I'm sure many of you know, convert into a product between the Fourier transforms. And that's useful because if we then do a calculation based upon that, we can literally cross out the Fourier transforms of the samples, the specimen spectrum cancels out. We'll ignore the fact, we can ignore the fact here that there's some issues here with zeros in the specimen spectrum. They are still there, but the later steps in what we're doing will allow us to cope with the fact that they cause problems. But if we just ignore that particular problem for now, it's very obvious that these will cancel out, and we actually end up with what we've called a pseudo PSF. We call it this because it's not really a point spread function, but it's in the same point spread function domain, and it contains the information about the ratios between these Fourier transforms of the PSF. So contained within this quantity here, the pseudo PSF, is the information we want about the aberrations, but we've removed the sample structure from the equation, almost literally removed it from the equation. Now, many of these other neural network solutions you will see are putting a huge amount of effort into trying to, using a lot of that neural network to ignore the specimen structure, to learn how to ignore the specimen structure. Now, why on earth would you do that when you can just do this type of operation here? So that's why we did this operation. And then what you realize is that most of the useful information in this pseudo PSF is actually contained right at the center, right around this spot you can see in the middle. And the rest of it's noise. It's noise or unuseful information. And so what we do is we take that and then realize that, well, we just need to base a neural network around the few pixels in the center of this. So we chose actually just the central 32 by 32 pixels. We could probably do fewer, but we just tried this and it worked. And then we designed a particular type of neural network, which was based around another piece of physical knowledge we have about this system. And this is similar to what we saw in the Wavelet transform thing earlier, where I said, well, we know that we have different scales of aberrations, that different features of the image will depend upon, will be affected differently by different sizes of aberrations. And this is effectively how this neural network has been structured, because we know that by using certain convolutional filters, we can kind of treat different parts of this neural network as accessing different scales of the image. So roughly speaking, we have the processes we're using here allow us to go between global features of the image down to local features of the image, and the image here being this pseudo PSF. And so, although there's not a perfect correspondence, we know that if we have different convolutions going on here, that we can actually relate this to the scales within the original images, and therefore we somehow mimic that idea we had in the Wavelet transforms, where we can access different scales of the images. And then we use that, pass it through the neural network, specially designed neural network, to enable us to estimate the Zernicke coefficients of the input. So more specifically, what it looks like is this. We have a stack of images we've taken from the microscope. Each one of these images will have had a different aberration applied. This, I've called it M here. This can be anywhere between 2 or many. I'll show you examples later where we've only used 2 images in this stack, and other ones where we've used a lot more. And then from that, we calculate all of these pseudo PSFs, and then this goes into we then do global max pooling to give you the first section of this. And this is a very unusual architecture. We've basically concatenated all of these neurons here in the neural network from each of these convolutional layers. So these convolutions down here are essentially what access the different scales of the image, and then the outputs from those goes into this one fully collected layer here, side by side, and then that goes through to another fully connected end and to the output. This is a bespoke neural network structure which has been designed specifically to solve this problem. And what's really interesting is that these weights here, Because we know there's a relationship between the information going into these neurons here and the spatial scales in the image, we know that the weights which are learned by the neural network process in this region here have physical relevance, and we'll use that later. I'll show you something about it. And that's quite unusual, because what we can not only is this design being inspired by physical knowledge of the system, we can also learn something about the physics of the system by looking at those weights, and we'll have a look at that later. This is very unusual. Normally, neural network architectures are black boxes. You don't look inside them, you just hope that the answer comes out correctly. Right, this still needs training. You can design this architecture and think this looks logical, but it still needs training, and the training data is very important as well here. So let me just briefly explain that. So you want to balance the learning in this across, so the data is realistic, so that it works across different types of microscopes. And as we said before, we need to essentially simulate the data, because we can't really get this out enough images, tens or hundreds of thousands of images out of a microscope. So we need to generate them in the computer. And so what we did was first of all, we used a diverse specimen structures, some of which, like these ones on the right, were actually downsampled real images from microscopes. So we knew we were getting the kind of typical structures you'd see in microscope images. And other ones, like this one here, were artificially created. And we used a whole combination of these, and we with various permutations of them as well in the training set, so that it was trained across a whole wide range of features it may actually encounter in a real microscope, not a simple set of one particular type of specimen. We then did various other variations of noise, of uncertainties in pixel sampling, because if you move from one microscope to another, there may be variations in the magnification slightly, which change this. So you don't want to overlearn the neural network based on one exact configuration. You need to make it flexible across many. And other things like higher order aberrations. So the data sets trained on took in all of these aspects to hopefully mean that later, once we trained this thing, it would work on different microscopes. And we're just going to compare different algorithms here. I'll just very quickly point out we're using two different conventional algorithms, just to give you a variety. One's called the 3n algorithm, the other one we've called the 2n+1 algorithm. Those of you who are familiar with this work will know about this. Those of you who aren't, don't worry about it. These are just two different conventional algorithms we've used in our previous work, which we wanted to use for comparison. We compared those two versions of the machine learned algorithms. One of them, which just used two acquired images, simply had the positive and negative amount of astigmatism, and another one which used the positive and negative images for each individual mode. Again, if you're not familiar with these methods before, don't worry too much about it. You'll see graphs later which just compare two of the new type of algorithm, two of the old type of algorithm, and you'll see very clear trends on this. So you don't really need to know the details behind this, but this does show that we were able to use this same overall architecture to cope with different styles of these types of algorithms, whilst using the same overall concept. So here's some examples, just so you understand the graphs which are going to come up. The red and the green one show the conventional algorithms. The blue and the grey ones will show the new machine learned algorithms. Okay, they're all based in a 2-photon microscope, and we're using a spatial light modulator to do a controlled experiment. It's actually introducing and correcting the aberrations in this demonstration. So you can see here on this graph here, we're just looking at beads. we see on the graph is the most important part here. Along the axis, you'll see the number of sample exposures we've taken in order to do the correction. It's different for the different algorithms, which is why they're differently spaced. And the fluorescence intensity along here tells us how well we've corrected this. And the key thing you can see here is that the two machine learning ones work a lot quicker than the conventional algorithms. And bear in mind, these were what we considered previously to be our good conventional algorithms. We've not picked the bad ones here. So you can see that the machine learned ones work better already. You can also see that was one particular example. If we take an ensemble of corrections, you can see here that even over the ensemble, the machine learned ones outperform the conventional ones. Also, if you look at the error bars, they're also a lot more consistent. We had a lot less consistent performance from the conventional algorithms as well. So this is paying off, this new design. We can go to, rather than just working on beads, we can look on arbitrary cell specimens. So some examples here where you can see again, in both of these examples, we've got totally different specimen structures. We've got better performance from the machine learned ones as well. And we can then look at what happens. Let's try making this more challenging. Let's turn down the laser power. So the image quality gets worse and worse and worse. And again, you see, in this particular case, they will perform better. In fact, some of the conventional ones barely perform at all. It's interesting in this one, we've got really low signal levels here. It's so low that if you look carefully, you can see a lot of just interference, electrical interference noise on the image. But even so there, we will then find out that one of the machine learned ones worked really well. The other one doesn't. Now, the one which doesn't work well is the one where we only took two images. And there's actually very good explanations why we'd expect that one to fail here. But the other method with taking more things still outperforms the conventional one. So, on all of these demonstrations here, we've seen that we can actually get a system now which outperforms our conventional algorithms. So the last part of this, I wanted to show this hint I gave you before about the fact that we could look at the weights after this neural network has been trained and optimized, the weights in this layer here, and find out if we can see anything about the physical information. And indeed, you can see something interesting here. If you look at the two examples, one of them is the 2-input machine learned one, and the other one is the 2n input, so which takes many more images to do the measurements. And what you see is that the distributions of weights in these different parts of this layer, you can see from the bar charts, are different. It's clear that we've got a big peak here in the upper level, and we're called M, which we are the maximum pixel values, and in the other one that's not the case. It's more distributed, and this makes sense because we would expect in the 2-input one, we'd expect it to be relying more upon the weighting, the fine detail in the images to be able to do the correction, whereas the other one actually contains more information in the relative brightnesses between the images, and that is indeed revealed through this. So we think that we can use this. First of all, it confirms our hypothesis about how this particular architecture would work. Secondly, we could use this to learn more about how these systems are learning, where the information is in these images. So this is a really interesting prospect for now to translate this towards other images, to translate this towards other types of microscopes. So to conclude, we've seen that by combining physical knowledge with special neural networks designs, we can come up with better algorithms which can take as few as 2 images to correct complex aberrations for extended objects. An interesting point about this is that the network complexity, and indeed therefore the training, is considerably simplified compared to previous methods. We are talking about perhaps 3 orders of magnitude fewer parameters in the neural network here compared to previous things that have been done in this area, which is obviously a significant difference. The methods outperform the conventional algorithms, and they're also got versatility to use the way we've trained these, and we're now actually deploying these onto other microscopes, which I haven't shown in this talk, but we now have results from this working on other microscopes too. And the concept is generalizable as well to other microscopes by changing. We just need to change the imaging model for the training data and then run exactly the same process, and it should work. So I'd like to thank you all for your attention. I'd like to, of course, acknowledge the main funders for this, which have been the European Trust and the Wellcome Trust and the European Research Council. And if you haven't looked already, please do go to this website shown on this slide, because there's a lot more information there about adaptive optics for microscopy.